This is isolation, characterization, and identification of malathion enrichment bacterial isolate Delphia lacustris from soil of the Cedar City orchard. First of all, why was this research performed? To understand that, a quick review of synapses is required. In between two nerve cells in a human body, there is a gap called a synapse. When one nerve is stimulated, it releases a chemical called a neurotransmitter. This neurotransmitter crosses the gap or synapse and fits into a receptor on the adjacent nerve cell. This results in a transfer of information. After this, the neurotransmitter leaves the receptor and is degraded by an enzyme in the synapse. Acetylcholine, a common neurotransmitter, has this chemical structure on the left. Nerve agents have a similar structure and are able to inhibit enzymatic degradation. A nerve agent occupies the site of this enzyme into which the neurotransmitter would typically fit. This prevents the neurotransmitter from being degraded. After the neurotransmitter relays the signal, the enzyme is inhibited and does not degrade the neurotransmitter. So the neurotransmitter remains and continues to stimulate the adjacent nerve cell. Acetylcholine, the typical neurotransmitter, is responsible for stimulating muscle constrictions and reducing heart rate. Therefore, this overstimulation results in paralysis and death to any persons exposed to sufficient quantities of these nerve agents. These fatal nerve agents are typically used as chemical warfare agents and insecticides, putting many civilians, soldiers, and farmers at risk. As seen in the previous slide, nerve agents are typically organic compounds that possess phosphorus. For this research, our goal was to find bacterial enzymes that can detoxify organophosphorus peptides and thus nerve agents. We could use these enzymes to protect farmers from back spray, clean up pesticide spills, and protect soldiers and other personnel from terrorist attacks. To find such an enzyme, we ventured to Cedar City Orchard. The pesticide commonly used at this site is malathion. Malathion has this chemical structure as shown on the right. It possesses a phosphorus atom and resembles the structure of these common nerve agents. The bacteria growing in the soil may have enzymes that allow them to degrade malathion and thus other organophosphorus nerve agents with similar structures. By taking samples of the soil and acquiring bacterial isolates, we hope to find bacteria with such enzymes. Again, we could use these enzymes to protect farmers from backspray, clean up pesticide spills, and protect soldiers and other personnel from terrorist attacks. To accomplish this, I first acquired soil samples from Cedar City Orchard and then transferred a small amount of soil to a tube containing carbon-free selective medium containing peroxone as the sole source of carbon. The purpose of the peroxone can be seen from its chemical structure. Peroxone is an organophosphorus molecule that resembles the structure of most nerve agents and pesticides. By providing this as the only carbon source, I hope to isolate only bacteria that could degrade peroxone and therefore chemical nerve agents as well. I incubated that initial tube for 1 week and then performed two more inoculation incubations into carbon-free selective medium containing peroxone in hopes to better isolate bacteria that could degrade peroxone. Subsequently, I transferred this liquid to an LB broth and incubated the mixture for 1 week. The mixture changed from transparent yellow to an opaque yellow, as seen here, indicating that bacterial growth had occurred. Since growth had occurred, the sample was ready for the next step. For this, I took a glycerol minimum medium plate and added a peroxone assay mixture with a cell spreader to continue to encourage only growth of bacteria that could degrade peroxone. I then performed a streaking for isolation of my broth culture onto the plate. After incubating for 1 week, this was the result. There were roughly 5 well-isolated colonies. I chose this one to focus on. As can be seen in the image, this colony was white in color and circular in shape. It was smooth and mucoid and about half a millimeter in diameter. Using the colony substance, I then inoculated an agar deep, another LB broth culture, and incubated for 1 week. As seen here, the resulting agar deep showed bacterial growth best at the top, but there was growth all the way to the bottom, indicating that the isolate was a facultative anaerobe. As seen in the next image, the culture changed from transparent to more opaque, indicating that the bacterial culture had grown successfully. So I smeared a mixture of my bacterial culture and nigrosine onto a microscope slide, and this was the resulting negative stain under 1000-fold magnification. The cells had singular grouping and were bacillus shaped. Each unit of the ocular micrometer indicates 1 micrometer. Thus, on average, they were about 1.6 micrometers long and 0.6 micrometers wide. I next performed a gram stain using the same broth culture. These were the resulting stain cells under 1000-fold magnification. As seen here, the cells are gram-negative. They were bacillus shaped and displayed singular grouping. They once again were on average about 1.6 micrometers long and 0.6 micrometers wide. They are the same size as the negative stain, indicating that this isolate does not possess a capsule. Since the gram stain indicated that my isolate was pure, I next began DNA isolation by using a microcentrifuge tube to obtain a cell pellet for my broth culture and then adding Triton X100 lysis buffer. These images show the solution after addition of the lysis buffer and then the solution after incubating the tube in boiling water. By comparing these two images, one can see that the solution had become more transparent, especially at the top, indicating that cell lysis had occurred. I next performed polymerase chain reaction and ran electrophoresis with the resulting DNA. This image shows the results of the electrophoresis. My band is on the far right. By comparing these results with a 1 kb sizing ladder from BioLine, one can see that the size was between 1,000 and 1,500 base pairs, roughly 1,300 base pairs, which aligns with the target small subunit ribosomal RNA sequence that was roughly 1,300 base pairs in length. The brightness best matches that of the 1,000 base pair band, indicating that it had a concentration of about 100 nanograms per band. Since these results indicated the successful application of the small subunit ribosomal RNA, I submitted the DNA to be sequenced. While I waited for the DNA sequencing, I inoculated a plate and a broth to perform a catalase test, an oxidase test, and a hanging drop motility test. Here are the results for each. In the catalase test, the gas was produced as shown by the formation of bubbles, indicating that bacteria is catalase positive. In the oxidase test, the filter paper turned blue after about 12 seconds after adding a bacterial culture, indicating that the bacteria was oxidase positive. Lastly, here's a video of the results of the hanging drop motility test. It may be difficult to see, but there is one bacterium that seems to dart across the view area in a specific path. This indicates that the isolate was motile. Upon completion of these assays, I attempted to identify the phylum to which my bacteria most likely belonged by using this dichotomous key for Bacillus gram-negative rods found at ABIS online. I followed this dichotomous key in this pathway, moving from straight cells to gram-negative rods to facultatively anaerobic to curved or vibrioid negative to oxidase positive. At this point, I realized that I needed to complete a test for nitrate reduction and acid from glucose before being able to complete the dichotomous key. I performed both tests, and these were the results. As can be seen in the image, the liquid for the acid from glucose was red preceding incubation just as it had started, indicating that the acid from glucose test was negative. The liquid in the nitrate reduction became red after testing, indicating a positive result. Moving back to the dichotomous key and moving along the acid from glucose negative line, we saw that the most likely genus is Ichinella. Upon this discovery, I went to the ABIS online encyclopedia, entered the Ichinella genus options, and found the species that best matched my biochemical assays, Ichinella corrodens. At this time, I received the DNA sequence of the sample I had submitted. Here is a sample of the resulting electropherogram produced. The peaks are narrow and sharp, indicating that the sequence was trustworthy. After reviewing the sequence, I fixed any miscalled bases and submitted it for blast search analysis, and the results produced two species. Firstly, Delphia lacustris, and second, Delphia titurohatensis. Both species had identical results with a max score of 1371, a query cover of 100%, and a percent identity of 100%. This image shows the alignment between the query and the top match, Delphia lacustris. As you can see, all 742 bases aligned, indicating a perfect match. Accession numbers are included for both species by their names. Evidently, the species determined from the dichotomous key and the blast results were different, so I returned to the dichotomous key to determine what might have gone wrong. I first followed the dichotomous key to determine where Delphia lacustris would be. I moved along the key following the path from straight rod cells to gram-negative rods to aerobic/microaerophilic to oxidase positive to non-yellow colonies, which my isolate also was, then motile, and landed at Bordetella, Chrysiomonas, Flavomonas, and Lecionella. One explanation for this very different path is that my species was not actually facultatively anaerobic, but actually an obligate anaerobe that can reduce nitrate when it runs out of oxygen, allowing it to grow identically to a facultative anaerobe in an auger deep. This would have caused me to erroneously follow the facultatively anaerobic pathway when I should have been following the aerobic pathway. Delphia lacustris is a nitrate-reducing species, and my isolate had a positive result from the nitrate reduction test, justifying this explanation. The blast search had only 2 results, and the ABIS online only had 1 that matched my biochemical assays, leaving me with 3 to compare. By comparing these resulting species, I determined that Delphia lacustris was the identity of my species, since it was a perfect small subunit ribosomal RNA sequence match and had the most in common with my isolate. As can be seen here, every characteristic matches between the two. Delphia lacustris is resistant to heavy metal toxicity and has been proposed for use as a biocontrol agent to protect against phytopathogens. Plants growing in soil that contains Delphia lacustris are less susceptible to root rot and wilt disease. Delphia lacustris can typically be found in water, on plants, and in soil. Let's see if I have to do it again.